Hi guys, welcome to StudyLib. I hope you all are doing well. In today's video, I will be doing class 12th chapter, that is Molecular Basis of Inheritance, which is totally based on NCERT as guided by NTA. Our content is liable and very beneficial for exam like CBSE board, CUET, NEET and AIIMS, ICAR, KCET and other medical entrance exam for 2023. So let's get started. First of all, you should be knowing very well that there are two types of nucleic acid present in a living system: RNA, ribonucleic acid, and DNA, deoxyribonucleic acid. RNA was first genetic material. RNA behave as catalyst. Catalyst is a substance which increases the rate of reaction. When RNA used as catalyst, it is called ribozymes. RNA can synthesize DNA. Nitrogenous bases of RNA are adenine, guanine, cytosine, and uracil. It can be single or double stranded. RNA is the genetic material of some viruses. It is chemically more reactive and structurally less stable. Now, let's talk about DNA. The sugar present in DNA is deoxyribose. In DNA, oxygen is absent from 2' on the pentose sugar, that is why it is called deoxyribose. Nitrogenous bases of the DNA are adenine, guanine, cytosine and thymine. It is always be double-stranded. Genetic material of almost all the living organisms. DNA is chemically less reactive and structurally more stable. Now, let's talk about the length of DNA varying organism to organism as per data given in NCERT. Bacteriophage 5X174 has 5386 nucleotide. Bacteriophage lambda has 48502 base pairs. Listeria coli has 4.6 into 10 to the power 6 base pair. Haploid cell of human DNA has 3.3 into 10 to the power 9 base pair. These data is very important for our competitive exam as well as your CBSE boards. Now, the structure of polynucleotide chain. A nucleotide is the basic unit of polynucleotide chain of DNA or RNA. Each nucleotide is composed of three components: a nitrogenous base, pentose sugar, ribose in case of RNA, and deoxyribose in case of DNA, and a phosphate group. Now let's have a proper understanding of this concept. When sugar is attached with nitrogenous base, then the combined structure comes out to be nucleoside. And when nucleoside is attached with phosphate group, the structure comes out to be nucleotide, which is the basic and structural unit of nucleic acid. Now, let's have a view on a very first component of nucleotide, that is nitrogenous bases. Nitrogenous bases are of two types: purines and pyrimidine. Purine include adenine and guanine. Pyrimidine includes cytosine, thymine and uracil. Thymine is present only in DNA and uracil is present only in RNA. A nitrogenous base is attached to pentose sugar by N-glycosidic linkage to form a nucleoside. When the phosphate group is attached to 5'OH of the nucleoside through the phosphodiester linkage, a nucleotide is formed. Two nucleotides are joined through 3' to 5' phosphodiester linkage and a dinucleotide is formed. When a numerous number of nucleotides are joined together, polynucleotide chain is formed. Now let's have a look on purines. Purines are large-sized nitrogen-containing biomolecules. These are 9-membered double-ring compounds. These contain 4 nitrogen in ring at first, third, seventh and ninth positions. Purines are both present in DNA and RNA. Purines include two types, adenine and guanine. Now let's have a look on adenine. Adenine is present in both DNA and RNA. Adenine is chemically called 6-amino-purines. Adenine have chemical formula C5H5N5. As you can see in the picture, the very important structure of adenine, including 4 nitrogen in first, third, seventh and ninth position and 9-membered double ring. The role of adenine in cellular respiration in the form of ATP. The role of adenine is also as cofactor nicotinamide adenine dinucleotide NAD and flavin adenine dinucleotide FAD. Let's have a look on guanine. Guanine is present in both RNA and DNA. Chemically, 2-amino-6-oxopurine. The chemical formula of guanine is C5H5N5O. As you can see in the picture, the very important structure of guanine. Guanine have two tautomeric form, the keto form and enol form. Guanine is first isolated from the excreta of seabird known as guano in 1844, which was generated as the source of fertilizers. Now, let's talk about pyrimidines. Pyrimidines are small-size heterocyclic nitrogen base. These are 6-membered single-ring compound. These contain 2 nitrogen ring at first and third position. Pyrimidines are 3 types: cytosine, thiamine and uracil. Now let's talk about cytosine. Cytosine is present in both RNA and DNA. Chemically, 4-amino-3H pyrimidine, 2-one. One means ketone group is present in cytosine. Chemical formula of cytosine is C4H5N3O. Now let's have a look on the picture, the very important structure of cytosine. It have amino group at 4 and keto group at second position. Now let's have a look on thiamine. Thiamine is present only in DNA. Chemically, thiamine is 5-methylpyridine, 2,4,1H, 3H dione. Dione means 2 ketone group is present in thiamine. Now let's have a look on the structure of thiamine. As you can see very well, 2 ketone structure is present and a single ring compound. Chemical formula of thiamine is C5H6N2O2. Thiamine is also known as 5-methyl-uracil. 5-methyl-uracil means a methyl group is attached on the uracil, it will become thiamine. It is derived from methylation of the uracil at 5th carbon position. Now let's talk about uracil. Uracil is only present in RNA. Chemically, pyridine, 2,4,1H, 3H dione. Dione means 2 ketone group is present in uracil or 2-oxy-4-oxy pyrimidine. Chemical formula of uracil is C4H4N2O2. As I explained in the previous slide that methylation of uracil gives thiamine, you can see in the picture very well that the methylation of uracil gives thiamine. Uracil show tautomeric shifts. The keto tautomers refers to lactam structure, while the enol tautomers refers to lactam structure. 5-fluorouracil is an anti-cancer drug. You can revise the whole structure from this. Pyridines include cytosine and thymine, and purines include adenine and guanine in the DNA structure. One end of polynucleotide chain contains pentose sugar with free OH at 5' end called 5' end, and the other contains sugar with free OH at 3' end called 3' end. The sugar and phosphate constitute the backbone of polynucleotide chain, and the nitrogenous base are linked to sugar moiety which projects from backbone. Now, let's have a view on some features of DNA. DNA is made up of two polynucleotide chains, where the backbone is made up of sugar and phosphate groups and nitrogenous bases project towards the center. There is complementary base pairing between two strands of DNA as you can see in the picture. The DNA strands are coiled in right-handed fashion and are anti-parallel in orientation. One chain has 5' to 3' polarity while the other chain has 3' to 5' polarity. The diameter of strand is always constant due to pairing of purine and pyrimidine. That is, adenine is complementary to thymine while guanine is complementary to cytosine. The distance between two base pair in helix is 0.34 nanometer. One turn contains 10 base pairs. Pitch is of 3.4 nanometer. Right-handed coiling means dextrocoiling. Angular divergence of DNA is 36 degrees. In between the nucleotide of double helical model of DNA, hydrogen bond is present. The ladder of DNA is made up of hydrogen bond and the backbone of DNA is made up of phosphodiester bonds. Both heliases of the DNA get separated from each other by melting and denaturation of DNA. Now, let's talk about double helical model of DNA. James Watson and Francis Crick in 1953 proposed the double helical model of DNA based on X-ray diffraction data produced by Morris Franklin and Rosalind Franklin and Irwin Chargaff rule of the base pairing. Now, let's have a view on Chargaff's rule. Chargaff rule states that the amount of adenine is always equal to amount of thymine and the amount of guanine is always equal to amount of cytosine. Adenine is joined to thymine with 2 hydrogen bonds and guanine is joined to cytosine by 3 hydrogen bonds. The ratio of adenine and guanine to that of thymine and cytosine is always equal to 1. The ratio is variable but it is specific for a particular species. For example, for human, A plus T by G plus C is equal to 1.52 and for Escherichia coli bacteria, A plus T by G plus C is equal to 0.97. This indicates that for lower organism, G plus C is greater than A plus T and for higher organism, the amount of cytosine and guanine is smaller than adenine and thymine. Now, let's have a look on central dogma. Francis Crick proposed the central dogma of molecular biology which states that genetic information flows from DNA to mRNA by the process called transcription and from mRNA to protein by the process called translation, as you can see in the picture very well. It is always unidirectional, except in viruses bidirectional because in viruses the reverse transcription takes place from mRNA to DNA. I hope you all find our video helpful. Thank you for joining. I will be coming back soon with some question based on the topic we covered today. That's all for today's video. Support us by like, share and subscribe our channel for more study related videos. Jai Hind, Jai Bharat.